Welcome back. Really, my desire to be creatively funny with my first PowerPoint on digestive system did not end as promising as I thought. So let's forget those puns and go back to our serious business, one bite at a time. Let's begin. This is now the second part B of chapter 23 on the digestive system. And we are going to be dealing mostly here with the role of the pharynx, or begin rather with, it's not only going to be this two, but we're going to begin with the role of the pharynx and the esophagus. So, food from the mouth would pass into the oropharynx, which is at the back, that part of the pharynx. The pharynx is actually a funnel shaped structure, uh, beginning at the back of the mouth all the way down to your voice box. So your pharynx passes from the mouth into the oropharynx, right there, that's the oropharynx, the yellow spot, and then into the laryngo. No, my bad. The oropharynx is the green part. The yellow part is the nasopharynx. So food comes from the mouth into the oropharynx, which is the green part, and then down into the purple area at the back of the trachea. And this is going to be helped through a reflex mechanism where the epiglottis would need to uh, the flap, which we know to be the epiglottis, will have to fold down and cover the entrance to the trachea. So the glottis, which is the slit-like opening, is going to be covered by the epiglottis. And so food would not have any way of going into your trachea, not unless, because you know that the reason why this reflex would pretty much like be entertaining um, reactions to two commands from the brain. One is by breathing, and that's actually the primary function of the epiglottis, to make sure that it stands up and would not be covering the trachea if air needs to go into the trachea. But it is also going to protect air from getting, rather water or any food material from getting into the trachea if need to, because we are eating, and therefore it's going to, this flap, which we know to be the epiglottis, will have to fold down and cover the glottis so food can be diverted at the back, which is in the esophagus. If we talk while we are eating, talking would need, or we use air when we are talking. So uh, the brain, or rather the epiglottis, would not know the difference between the need for the air to survive, or the need for the air, our need for air when we talk. The thing is, we just need air, and so it's not going to fall down when we are eating and talking at the same time. It's not going to fall down to protect the trachea. It's going to stay up because its primary function will be to make sure that the air goes through in here. When air is needed, uh, according to the brain, you are talking and therefore you need air so you can actually project your voice. So it stays up. And that's when we pretty much would uh, um, experience gag, or some food actually gets in there, and we are going to be choking. So we'll have to avoid that and prevent that kind of accidents by making sure that we swallow our food before we start talking. So your pharynx, being that it is an extension, the back of the buccal cavity, being that it's an extension of the oral cavity, would be lined with stratified squamous epithelium and with mucus producing glands. It would have external muscle layers consist of two skeletal muscles. The inner layer of muscles would run longitudinally, and the outer pharyngeal constrictors, which is actually circular muscles, would encircle the wall of the pharynx, which would help in the powerful muscular contraction that is needed during the pharyngeal phase of swallowing. And here is actually a cross section of the esophagus, which will give us an idea on what the esophagus actually is made of. So, because it is actually it's a tube that would have heavy traffic as we are swallowing food, so food is constantly like passing through it. We do not expect the esophagus to have only a single layer of epithelium. We expect it to have multiple layers, and therefore the esophagus is just like the pharynx, and the oral cavity is going to be lined with stratified squamous epithelium with and the underlying mucosa is made up of areolar connective tissue. It would have also muscularis externa, which would be made up of the circular, an inner circular, and an outer longitudinal layer. And since it is not inside or within, the location of the esophagus is not in the abdominal cavity, you do not expect the esophagus to have any visceral peritoneum. It would have adventitia. That's actually the cover of the esophagus. So your esophagus pretty much is a flat muscular tube that is actually located at the back of the trachea. It runs from the laryngopharynx all the way down, connects into the stomach. The esophagus is collapsed when not involved in food propulsion. So, uh, it pierces the diaphragm, right? That section there, that's your diaphragm right there. It pierces the diaphragm at the esophageal hiatus and joins the stomach. Right there, it joins the stomach. It joins the stomach at the right there by the cardial orifice or the gastroesophageal sphincter. It's the gastroesophageal sphincter um, surrounds the cardiac orifice. This is actually the orifice is the opening itself. The sphincter is the muscle. It's a circular muscle that surrounds the orifice, which keeps the orifice closed. It is supposed to be closed whenever we are not swallowing. The only time that the orifice should be opened is when we are swallowing food. So it is always closed so that um, whenever we're not swallowing food and there's food there that needs to be digested, then you know that one of the um, one of the things that the stomach will do will be to produce the substances that is necessary for it to digest the things that it should digest. And one of the things that is being digested in the stomach is actually actively is protein. And when protein is digested in the stomach, it would need um, pepsinogen, which is um, the enzyme, the protease enzyme that would be digesting protein. Now, for this protease enzyme to be active, it is in an active form, which is pepsinogen. It needs to be activated to its active form, which is pepsine, and you require hydrochloric acid for pepsinogen to be converted into pepsine. That's why the contents of the stomach is very acidic, most especially if it is digesting protein. Now, if that particular opening is not going to be closed by the gastroesophageal sphincter, then the stomach, which is actually really very heavily which is actually heavily muscular, when it contracts, it's going to be contracting forcefully and would push the food all over, and it could, even if it's only a small amount, that highly acidic substance, which we call the gastric juice, will pretty much like go back into the esophagus and will burn your esophagus, and that's actually what you call the gastroesophageal um, uh, gastroesophageal disorder, GERD. It is called the gastroesophageal reflux disease, or GERD, and can be caused by excess food or drink, maybe extreme obesity, pregnancy. Running can also be caused by hiatal hernia, and anything that is um, structurally going to lead in the what do you call this, in the inability of that particular sphincter to close, will cause GERD. And this could lead into what you call esophagitis. Esophagitis is inflammation of the esophagus. The esophagus has four layer. Okay, it would have also the esophageal mucosa, which is a stratified squamous epithelium, and it changes to simple columnar right when the moment it reaches the stomach. So between the esophagus and the stomach now becomes an organ that is not only a passageway. The esophagus is only a passageway. It is necessary for the epithelium of the esophagus to be stratified. But the stomach, it is not necessary. There's actually one very important function of the stomach that would not require the presence of stratification in the cover of the stomach. When the stomach has too much stratification, then the one function of the stomach will not be possible. The stomach needs to pump out substances as part of the digestive juice of the stomach. Now, stratification would not be a good uh, a good way by which an by which an organ um, that would need to pretty much like pump a substantial amount of gastric juice would be. It would need a good source of an immediate source of um, substances when it needs to really like start digestion. And by immediate, I mean it cannot be at the deep inside the stomach. It has to be like within the surface of the stomach. And when it pumps out, it secretes the substance that it should be ready it should be available, readily available for the use of the stomach to digest food. So um, we have only a simple layer of columnar tissue that lines the stomach. From the esophagus, which is stratified squamous, it becomes simple columnar, and the esophageal glands are part of this simple columnar um, layer. And it's out this esophageal. There are also lots of esophageal gland in the submucosa, and they secrete mucus to aid in the movement of the bolus. Remember, when food lands in when food is mixed with the saliva, you call it bolus. But when bolus is swallowed and gets in contact or touches gastric juice of the stomach, you don't call it anymore bolus, you call it chyme. So, um, in order to help in the movement of the chyme in the stomach, uh, there's going to be like a large or a good number of muscularization in the stomach. The stomach itself does not only have one longitudinal and one circular muscle, like the tubes of the esophagus and the small intestine, it would have three layers. It will have um, oblique layer, which is the extra layer in addition to the longitudinal and the circular layer. So your stomach can actually move three-dimensionally. It's not only going to be like peristalsis or segmentation. The stomach can actually move because it is large and heavy, and it can move actually in many directions because it would have um, longitudinal, circular, which is perpendicular to one another, and then right in the what do you call this, right at 45-degree angle with them will be the oblique muscle. And it is covered with adventitia instead of a serous membrane. The pharynx and the esophagus, you could see right here, are conduits to pass food from the mouth to the stomach. From the mouth to the stomach, it goes right through the esophagus. The major function of both organ is propulsion that would start with swallowing or deglutition. Deglutition involves the coordination of 22 muscle groups and two phases in its swallowing. The first phase is what you call, this is actually the picture of the buccal phase or the buccal phase. The buccal phase is the voluntary contraction of the tongue. The second phase is, um, this thing doesn't want to work. The second phase is actually your uh, pharyngeal esophageal phase. And immediately after the buccal phase will be your pharyngeal esophageal phase. This is the involuntary phase that primarily involves the vagus nerve, which is controlled by the swallowing center in the medulla and the lower pons. Then you have the third phase, which is the pharyngeal, the continuation of the pharyngeal uh, esophageal phase. The constrictor muscles of the pharynx contract, forcing the food into the esophagus inferiorly. The upper esophageal sphincter would contract after the food enters. Then you have the fourth phase, which is the peristalsis, the movement, rather the peristaltic movement of the esophagus. It moves the food through the esophagus all the way down into the stomach. And you could see here the food is actually moved into the stomach through the peristaltic movement of the esophagus. The gastroesophageal sphincter, which is actually the one that guards the opening or the orifice going into the stomach, will need to open in order to allow food to get into the stomach. After the food enters the stomach, the sphincter will need to close immediately, preventing regurgitation or preventing food from getting back um, into the esophagus. Most especially if it has already landed here, the bolus plus the gastric juice is now your chyme. The chyme is very acidic. Your chyme does not need to go back into the esophagus or else it's going to give you pain. And here is actually an introduction to some names that I would like you to know and recognize so that you will understand exactly what we're talking about. And these are the intraperitoneal membranes. There are names, even of fatty materials, that you would need to know to pretty much like significantly understand the positions of the organs in the abdominal cavity. For instance, your abdominal cavity, the abdominopelvic cavity, is lined by parietal peritoneum, and the parietal peritoneum in this picture is actually the dark blue line. The yellow, um, mild yellow line is actually the cavity itself. Cavity is lined by the dark blue line right there, and that is your uh, parietal peritoneum. The visceral peritoneum are the ones that are covering the organs themselves. And you could see the visceral peritoneum here as the red. Can you see that? Those red. Let me just put on my marker. Okay. My red, okay, that's actually your red. The red cover of each of those little organs there are actually your visceral peritoneum. Now, the peritoneal cavity is that particular space that is the light blue color. Now, from the visceral peritoneum, okay, let me see, let me give you, okay, from the visceral peritoneum, which is the one that's covering the organ. Let me show you this organ here. This organ right here is covered by a visceral peritoneum. That's there, right there, and is suspended into, let's say, the dorsal body wall. It is actually suspended by a mesentery. Now, because this is actually like, well, I would think that it is actually the transverse colon. So this is the transverse colon. So it's a colon. So this type, this mesentery here is named the mesocolon. So your transverse colon is actually like connected to or suspended in to the dorsal body wall via a mesocolon, which is really very fatty. Now, the stomach. Take a look at the stomach right above the transverse colon. You have the stomach. It is also covered by a visceral peritoneum, but is actually suspended to another organ, connected to another organ. This organ here, which is the stomach, is connected to the underside of the liver right there. And this particular connection is an omentum. An omentum is just like mesentery, all right? Um, this particular omentum is connecting the stomach, the lesser curvature of the stomach, to the underside of the um, underside of the liver, and you call this the lesser omentum. And when we get into a better picture of the stomach, I will show you where the lesser omentum and how the lesser omentum looks like. Now, on the greater curvature of the stomach, you have another mesentery type or another um, intraperitoneal membrane, another omentum, and this omentum drapes over and is actually like suspended and draping over and is connecting to the transverse colon into the inferior line on the inferior margin of the entire transverse colon. This is actually what you call the greater omentum. The greater omentum is loaded with fat, and the purpose of this is to create some sort of a significant protection uh, to the through the anterior portion of the abdominal cavity, which pretty much doesn't have any bone. So you have your thoracic cavity in the front that protects the heart, and your heart is actually really bounded by all over by bones, but the front part of your belly isn't, and the front part of your belly needs to have protection other than muscles, and therefore one of the protection is actually your greater omentum. Greater omentum connects the stomach and the transverse colon as like an apron. You could see that the greater omentum goes down from the stomach down and then goes up again and joins the transverse colon. It is like an apron. Then you have also the large intestine, okay, which is actually suspended to the dorsal body wall by what you call a mesentery. This is actually what you call the mesentery proper. All right. Uh, did I miss anything? And somewhere here in the back, you will find why they did not put it here, but somewhere here. Oh, I know why. Because this is actually a cut that is along the midline, which is actually the mid-sagittal section, and therefore um, you will not see the kidneys, because the kidneys are located like parasagittal. They are actually on the side and are not found in the midline. But you will see some of the ones that is colored light green, and they are the ones that are what you call retroperitoneal. They are not inside the peritoneum. Your pancreas, your duodenum, which is actually the beginning of your small intestine, is retroperitoneal. The rectum as well as the urinary bladder are also retroperitoneal. And then the two kidneys are also retroperitoneal. So the rest of them are intraperitoneal. So here is your stomach, and you will pretty much like us. You need to significantly know the parts of the stomach because this is going to be in your exam in the lab next week. Hmm. Yes, it's going to be part of your lab because this is. mm, Now, I do not know if it is actually going to be part of the coverage of the lab, but you'll have to check on the announcements. Okay, so your stomach is a temporary storage bank that starts chemical breakdown or protein digestion. It converts the bolus into a paste-like chyme. Once your gastric juice acts upon the bolus, it becomes, because it's very acidic, it becomes paste-like. Now, an empty stomach has 50 ml volume, but can expand up to 4 liters. A person really, when I was watching those great food binging contests, such as the Nathan hot dog eating contest at Coney Island, New York, I could not imagine how they can actually handle that much hot dog, and that includes the bread and some water and also the hot dog itself. Now, the stomach can actually expand up to 4 liters, and you could just imagine. When empty, the stomach mucosa forms many folds, which you call the rugae. This is actually the fold of the stomach. You could see it right here, and that's actually what you call the rugae. So let's take a look at the major regions of the stomach. The cardia, or the cardiac part. This is actually the cardiac section right there. Okay, the cardia is called the cardia because it is closer to the heart. And this cardia part is surrounding the cardial orifice, the orifice or the opening into the stomach. And the fundus is actually the dome-shaped region, that dome-shaped region, which is actually beneath the diaphragm. The body of the stomach, this part here, is the mid portion, and the pyloric part is the one. This part section right here, the pyloric part, is the one that is closest to the pyloric end or the pyloric sphincter, and the antrum of the stomach is this antrum right there. This is the antrum of the right there. That is the so sorry. I cannot control my pointer. The pyloric antrum is actually the area that narrows into the pyloric canal. This is now your pyloric canal that terminates into the pylorus, which is continuous with the duodenum through the pyloric valve. The pyloric valve is the sphincter that controls the emptying of the stomach. The pyloric valve is what I call is also um, uh, well, I remember my professor when I was studying this saying that the pyloric valve is a lazy valve and it needs to be lazy. That is actually its function to be a lazy valve, simply because it is only allowed to release food into the duodenum in spurts, about 3 ml. Could you imagine how much is 3 ml? About 3 ml per minute only. That is exactly how slow the emptying of the stomach to the duodenum should be. Why? Because the stomach carries a lot of acid in it, and it cannot really like dump that very acidic chyme immediately into the duodenum, or you're going to create like duodenal ulcers there, because you're going to burn the um, um, the mucosa of the duodenum because of the acidity of the chyme. You'll have to make sure that you slowly empty the partially digested food into the duodenum. And then in the duodenum, you have a lot of bicarbonate ions that is being produced by that is actually part of the pancreatic juice that's going to be secreted there. And the duodenum itself has um, walls that is rich in that is actually rich in that has cells. The walls of the duodenum has cells that could actually produce big time or a large amount of basic bicarbonate substance um, that would significantly neutralize the acidity of the chyme. So your stomach has a lesser curvature. This is where the lesser omentum is connected and is going to be connecting or anchoring the stomach to the lower surface of the liver. And then it has a greater curvature. This is the greater curvature is where your greater omentum is connected. These mesenteries will make sure that the stomach, no matter how actively the stomach is actually moving when it is digesting food, will have to stay right where it is supposed to be. This is your stomach. It's a lesser omentum of the stomach, and this is the greater omentum of the stomach, and this is a cadaver. That's where your spleen is located. Okay, let's move on. There you go. This is your lesser omentum, and this green portion here. This is not the real color of the lesser omentum. Uh, it is just colored green so that it could be highlighted. So the green part is actually the uh, lesser omentum, and this part here, which is really loaded with fat, that is connected to the greater curvature of the stomach, is called the greater omentum. It blends with the mesocolon, which is the mesentery that anchors the large intestine to the abdominal wall. It contains a lot of fat deposits and lymph nodes. Here's another picture of the stomach showing you the innervation of the stomach. The autonomic nervous system supplies the stomach, and you have sympathetic fibers from the thoracic splanchnic nerves that are relayed through the celiac plexus, and the parasympathetic fibers are supplied by the vagus nerve. The stomach walls contains 4 layers. However, the muscularis and the mucosa are modified. The muscularis externa has circular and longitudinal smooth muscle layers, as well as an extra third layer, which I have already mentioned, the oblique layer. So let me see. Okay, your longitudinal layer is the outermost, just like you in the esophagus as and in the small intestine. The outer layer is always the longitudinal layer. Next to the longitudinal layer is actually running longitudinal, parallel to the axis of the tube. And then next to that is a second layer, middle layer, which is the circular layer, which is parallel to the longitudinal. So this one here is running cross-sectionally. Then you have an oblique layer, which is actually neither longitudinal nor parallel. It actually runs at 45 degrees to both. We call that the inner oblique layer. The third layer that would allow the stomach to move three-dimensionally. So together, these smooth muscles would allow the stomach not only to churn and mix and move, but also to plummet it and move it pretty much with great physical breakdown and ram it into the small intestine. But of course, you should have the pyloric sphincter that is supposed to be closed really tightly, and it's not going to allow really food just to be emptying fast. It needs to be emptied out very slowly into the duodenum, right through the duodenum. Okay, so here is actually the mucosal layer, which is also modified and consists of simple columnar. You could see that it is actually simple columnar. Your stomach is actually is also like wrinkly inside. It forms lots of folds, which you call the ruga. The ruga increases the surface area of the stomach for absorption and for secretion. That will increase the number of cells, which is actually here you could see the cells of the mucosa of the stomach that is producing the gastric juice. Now, the stomach cells pretty much are made up of mucous cells. They secrete two-layer coat of alkaline mucus. The surface layer would trap the surface layer would trap the bicarbonate-rich fluid layer that is beneath it. The mucosa surface is dotted with gastric pits. If you're going to look on the surface of the stomach, you'd see that the surface of the stomach seemingly would have holes. Can you see those holes right there? They are what you call gastric pits. And if you continue right through, the gastric pits is really nothing but the neck that is actually continuous with what do you call this? Um, some sort of like a not really a container, but a long line of secretory cells right inside producing gastric juice lights along the wall of the stomach. This is a close the gastric pit, showing you the gastric pit. You have the different cells that is actually in the glandular area of the stomach, and you have different glandular cells, the glands in the fundus, and as in the fundus of the stomach, the curvature in the stomach, and the body of the stomach produces the gastric juice. These glands would include the secretory cells. And first of all, you have the mucous neck cells right there, okay? And you have also the parietal cells, the ones that are in blue are parietal cells. Then the ones at the bottom are what you call the sheep cells, and the green one is what you call the enteroendocrine cells. So, your parietal cells, let's start with the parietal cells. What is actually the purpose of your, well, first of all, let's start from the top. The mucous neck cells, these cells secrete thin acidic mucus of a known function. Really, the only function of acid itself is to make sure that the acidic environment of the gastric juice is available for the conversion of pepsinogen into pepsin, which is the enzyme that is necessary for the digestion of protein. Now, parietal cells, that's your parietal cells. These are cells that would be producing hydrochloric acid. Hydrochloric acid gives the pH of the stomach, or lowers down the pH of the stomach to about 1.5 to 3.5. It is This pH is enough to denature the protein and to activate pepsin from pepsinogen. It breaks down plant cell walls and would kill many bacteria. The parietal cells secretes what you call the intrinsic factor. The intrinsic factor is a glycoprotein required for the absorption of vitamin B12 in the small intestine. Now, the cheap cells, that particular purple part there, secretes pepsinogen. Pepsinogen is a precursor that would need to be converted into the active form, which you call pepsin. So pepsinogen is an inactive enzyme, and it needs to be activated through pepsin by HCl. It needs to be activated at a very low pH of about 1.5 to 2. Okay, so once it is activated, it gets activated as pepsin, which is a protease. The cheap cells are also, these cheap cells are also secreting gastric lipases. Um, Gastric lipases are not really very much. It secretes only about 15. It is very little, and therefore it can only be breaking down about 15% of the fat that we eat. The enteroendocrine cells, the green ones at the green part, which is all the way at the posterior end of the gland itself, secretes chemical messengers into the lamina propria. It acts as paracrines, and paracrines are um, hormone-like or substances like hormones that would um, stimulate or talk to its neighboring cells. Okay, so it secretes serotonin and histamine. The cell also secretes hormones such as somatostatin, which also acts as a paracrine. And gastrin itself is also being secreted by these enteroendocrine cells. So now you have a more closer view of the distal portion of the gastric pit, and it shows you the parietal cells. The parietal cells producing HCl, and HCl is needed by uh, in the conversion of pepsinogen to its active form, which is pepsin. And pepsinogen is produced by the cheap cells. Now you will need to know which cells are producing these enzymes. Parietal cells producing HCl. The cheap cells secrete pepsinogen, but this pepsinogen is inactive, needs to be activated to pepsin so that pepsin can break down protein. Then you have your enteroendocrine cells. These are the ones that secretes the hormones and the paracrines of the stomach. So, the mucosal barrier. I have just pretty much like given you um, big information that would pretty much like result, or that could probably like culminate into a big question. So, if pepsinogen gets converted into pepsin, and pepsin is a very powerful protease. How come this protease is not digesting the walls of the stomach? Well, the reason for this is because we have a mucosal barrier, and that's actually your mucosal barrier right there. The mucosal barrier pretty much would prevent uh, the destruction of the stomach wall. The harsh digestive condition requires protection, and the protection is actually the mucus that is produced as part of the mucosal barrier. there is a thick layer of bicarbonate. You could see that those bicarbonate rich mucus that is being produced by the mucous neck cells right there. And there's actually like tight junctions along the epithelial cells, the mucous neck cells, which prevents the juice, this juice that's actually being produced by these, wild gastric juice producing enzymes. They have actually very tight junctions in between the epithelial cells that prevent the juice from seeping through and destroying the rest of the protein of the stomach. But of course, the bicarbonate rich mucus is actually the one that helps and protects the walls of the stomach. The damaged epithelial cells, and it's actually like imminent that there could be damages to epithelial cells because of that. Yes, they are quickly replaced by division of stem cells. The surface cells are replaced every 3 to 6 days. Gastritis is an inflammation caused by anything that breaches through the stomach mucosal barrier. Peptic or gastric ulcers can cause erosions in the stomach wall. If erosions perforate wall, it can lead to peritonitis and hemorrhage. And most ulcers caused by bacterium, which is the Helicobacter pylori, can also be caused by non-steroidal inflammatory drugs such as aspirin, okay, or some other drugs that we are taking and that is actually harmful to the mucosal wall and can cause gastritis. A long time ago, before you were even born, we thought that ulcers are caused by stress. It is actually exacerbated by stress, but it's not really caused by stress. The one that causes ulcers is actually a bacteria, which we call the Helicobacter pylori. Uh, Something like maybe 4 decades ago, or 5 decades ago, um, maybe 5 decades ago, even before your parents probably were born, there were two Australian scientists who who experimented on rats on the study of ulcers, and they realized that it's not stress that's actually causing the rat to develop ulcer, and they realized that it's actually a type of bacteria which they named Helicobacter pylori. Nobody would believe them, and for for some reason one of them decided that they needed to really um, prove to the world that their experiment is true. So one of them, um, one of them would agree on taking the bacteria itself and develop the ulcer, while one of them would be holding an endoscope and would pretty much like be showing the world that there's actually ulcer forming, which they did. They were able to dangerously, and no matter how dangerous it is, their their what I call is their style and of proving to the world their point was um, pretty much like very dramatic and was not really approved by the scientific community, but they proved their point. From then on, we know now that the one that produces ulcer is not stress, but H. pylori. So let's take a look at the processes carried out by stomach. Now, the stomach itself carries carries out breakdown of food. It serves also as a holding area for food. It delivers chyme to the small intestine. It denatures the protein by the hydrochloric acid that it produces. And then pepsin, which is actually the activated pepsinogen, carries out the enzymatic digestion of proteins. And then, just on the side note, I want to make mention milk protein, which we call casein, is broken down by renin in infants, which results in a curdy substance. That's why whenever babies would burp out and throw up the milk because of overfeeding, you could see that curdling milk that is actually being curdled out. This is actually brought about by renin. It's actually an enzyme that babies produce as part of their gastric juice that would break down milk. Then you have lipid soluble alcohol and aspirin, which it's noted that are actually absorbed into blood through the stomach. Only stomach function essential to life. The only stomach function that is essential to life. All of them is actually only pure digestion and a little bit of absorption. But the only stomach function that is essential to life is the secretion of the intrinsic factor by the parietal cells. The intrinsic factors is actually necessary for the absorption of vitamin B12. And vitamin B12 is needed for red blood cell maturation. The lack of intrinsic factor will not allow for many red blood cells to mature properly, and therefore it can cause pernicious anemia. The treatment for pernicious anemia usually is vitamin B12 injection. The gastric mucosa secretes 3 liters of gastric juice per day and is actually regulated by the stomach neural mechanism through the vagus nerve stimulation that increases its secretion. The sympathetic stimulation of the stomach would decrease secretion. Sympathetic is actually the fight, flight, fight response. And so if we are always worried and if we are under stress, that is actually sympathetic innervation. And so your mom and dad would always say, stop worrying or else you're going to have indigestion, which is true. You are going to have indigestion because your stomach will slow down whenever you are worried. But when you are relaxed, then your vagus nerve is actually free to release the parasympathetic stimulation and it's going to promote digestion. The hormonal mechanism in the stomach, just like what I mentioned to you, is gastrin, the secretion of gastrin by the enteroendocrine cells. Gastrin stimulates hydrochloric acid secretion by the stomach, and gastrin antagonist hormones also is secreted there in small by the and gastric antagonist hormones is secreted by the small intestine. Okay, now we're going to get into the neural and hormonal mechanism that regulate the release of gastric juice. So, the regulation of food that gets into the stomach or the food that we put into our mouth and pretty much gets into the stomach does not start when we put food in the stomach. It starts even before we put food in the stomach. When our stomach is empty, we feel hungry. Even if we don't feel hungry, there are some things around us or things inside our brain, our thoughts, that could actually trigger hunger. In the cephalic phase, the mechanism of the mechanism that releases or activates the gastric juice are actually of 3 phases. The first one is what we call the cephalic phase. Let me see my pointer. That's your cephalic phase right there. This cephalic phase is controlled by the brain. This is actually the things that govern our thoughts about food. So even if we're not hungry and if we're driving around and we saw McDonald's and we saw that Big M and that clown, all of a sudden we feel hungry. Why? Because now you see something that represents food, even if you don't smell food, you don't see food. You saw Ronald McDonald and you see that big M, you know that there's food there and you feel hungry. That's actually already a cephalic phase. Or cephalic phase could be also like all of a sudden you were walking and you smelled like food being cooked, garlic, and you love the smell of that, or onions. There are you love home cooked meal and you smelled it all of a sudden. You don't even see it, but you smelled it, then you become hungry. That is part of your cephalic phase. Or let's get it closer. You're walking and you saw food right there and a picture of food right there because you're just very close to a China buffet. You don't even smell it, but you saw the picture and it made you hungry. That is cephalic phase. Did food land on your stomach? No, food did not land in your stomach. You just thought about it, you saw it, or probably you smelled it, you thought it, and it made you hungry. So this is actually the cephalic phase, which is nothing but the stimulatory events, the sight, the thought, and the stimulation of the taste and the smell receptors that pretty much like is coming from the cerebral cortex is brought down to the hypothalamus and the medulla oblongata right through the vagus nerve and is going to send information to the stomach, and that's going to increase the production. It's going to increase, it's going to promote the stomach to get into the second phase, which is the gastric phase. In the gastric phase, the stomach distends, activates and the stretch receptors. This means in this particular phase, food pretty much is already taken in. So you're not only looking at the food and smelling food here, you really got hungry and you ate food and you swallowed it. Now you are now in the gastric phase. You're now going to be continuing to eat food. The stomach walls become distended. It activates the stretch receptors. Pretty much, this will activate the movement of the stomach. Food chemicals, especially the peptides and caffeine, also rising pH would activate the what you call the chemoreceptors or the G cells, and then the gastrin will be released into the blood. This will increase stomach activity. So your stomach now has food inside it and it will continue to churn. It will continue to churn until some parts of the chyme reaches the distal end, which is actually the pyloric end, and it is now going to stimulate the opening of the um, um, the duodenal, the pyloric sphincter right here, and it will release small amounts of food into the duodenum. The moment that first drop or a few amounts of chyme is actually dropped into the duodenum, the duodenal cells, the enteroendocrine cells of the duodenum, is now going to send off information to pretty much like uh, to the liver to release the enterogastrones, your secretine and cholecystokinin. Remember, the distension of the duodenum and the presence of small amount of acidic hypertonic chyme in the duodenum will release this enterogastrone, and this will now immediately, it's an immediate effect. This is now going to have a what do you call this? An inhibitory effect on the stomach itself. So during the first phase of your eating, the first 20 minutes of your eating probably, you're just eating food, but the moment that first drop of chyme gets into the duodenum, you're now going to release some inhibitory enzymes or inhibitory hormones that's going to stop or that's going to inhibit the movement or the motility of the stomach. This is when you feel you are full. And it happens like clockwork. For as long as your stomach has the ability to pump small amounts of chyme there, that small amount of chyme will make you feel full because that small amount of chyme will help in the um, release of the enterogastrones. And once your enterogastrones, secretin and cholecystokinin are there, it's going to inhibit the release of gastrin. Once gastrin is inhibited, then the motility of the stomach is slowed down. And that's a reason why, after certain time, um, you don't feel anymore hungry because your cholecystokinin and secretin is now inhibiting your stomach motility. There are also other things that could inhibit the events of the stomach. For instance, anything that is cephalic in nature or cerebral in nature um, could actually lead to a loss of appetite, such as depression, okay, or some problems, something that is actually stressing you out, or anything that is excessively acidic, that is less than pH 2, will pretty much like inhibit the G cells also, and will inhibit gastrin, and pretty much emotional stress will also inhibit the movement of the stomach, besides the fact that we have the presence of food in the duodenum. But the presence of food in the duodenum is like clockwork. It happens about the first 20 to 25 minutes of you eating. So if you're going to slowly eat your food and pretty much like chew your food slowly, very slowly, then in the next 20 minutes, the first drop of chyme will make you lose your desire to eat more because the movement of your stomach will slow down. So when our parents would say, slowly eat your food and you're going to lose weight, darn, that thing is true. That thing is true. You want to lose weight? Don't eat food too fast. And here is actually a cheat sheet, the hormones and paracrines that act in the digestion. I'm hoping that this is going to help you. Make sure that you study this, guys. So let me try to discuss a little bit the mechanism of hydrochloric secretion by the parietal cells. The parietal cells, those are your parietal cells right there, the blue one. And this big thing here is your parietal cell. The parietal cells would pump hydrogen ion from carbonic acid, from the breakdown of carbonic acid into the stomach lumen via the hydrogen potassium ATPase proton pumps. Okay, this is actually your ATPase proton pumps. You could see that from the potassium protein, rather the positively charged ions are being produced and they are actually being pumped in. So you have positive ions that are actually being circulated in your stomach. The hydrogen ion is pumped into the stomach lumen. The bicarbonate ion is exported back into the blood via the chloride and bicarbonate antiporter. And the resulting increase of bicarbonate ion in the blood is going to leave the stomach and is referred to as the alkaline tide. See that? The bicarbonate ion leaves the stomach and gets into the bloodstream. You call that the alkaline tide. As it leaves the bloodstream there, now there's an antiport, which means this bicarbonate ion leaves, then that would allow the chloride ion to enter, and then outside and into the stomach lumen, you are going to be put together with the hydrogen ion. The chloride plus the hydrogen ion is going to produce HCl. That's exactly how HCl is actually formed. Okay. That's a good way of doing that. I did not follow that particular stages right there. It's a good way of doing that. I did not know it's going to do that, but it's the same thing. All right, let's take a look at the response of stomach to filling itself. The stomach wall stretches. It stretches to accommodate the incoming food, and two factors can cause the pressure to remain constant until 1.5 liter of food is ingested. Your stomach has a capacity of taking in about 1.5 liter of food, and it gets to be bigger as you practice on eating a lot of food. Your stomach can actually get bigger too. So, the first phase is actually, well, you have swallowing, and then you have feeling, and then there's actually what you call receptive relaxation. Receptive relaxation is a reflex mediated relaxation of the smooth muscle in the stomach that's coordinated by the swallowing center of the brain. Then you have what you call gastric accommodation. The gastric accommodation is the intrinsic ability of the smooth muscle to exhibit stress relaxation response, which enables hollow organs to stretch out really without increasing tensions or contractions. Gastric contractile activity is actually the peristaltic waves that you could see here, that moves towards the pylorus at the end at the rate of about 3 peristaltic activity per minute. And that's really a lot. Then you have also what you call the basic electrical rhythm that is set by an enteric pacemaker, a pacemaker that is actually in the interstitial cells of we call it the interstitial cells of Cajal. These pacemakers are linked by gap junctions so that the intermuscularis contracts and the distension and gastrin increases the force of contraction. So the more food you actually take in, the more gastrin is produced, then the motility of the stomach through peristalsis is also increased. So how is well so how is gastric emptying going to happen? Well, the duodenum itself can prevent overfilling by controlling how much chyme enters there. See, the duodenum is not going to allow too much chyme to enter into its realm because the duodenum has a very basic environment. It is not like the stomach where it could actually handle very acidic chyme. In the duodenum, the environment of the duodenum starts from pH 7.1 to about pH 8. And you don't want the duodenal cells cannot withstand very acidic chyme, or else it's going to kill a lot of the cells. Duodenal receptors respond to stretch and chemical signals. The enterogastric reflex and the enterogastrones would inhibit gastric secretion and duodenal filling. It would slow down duodenal filling. That's why I told you one of the slowest sphincter that can move things through the gastrointestinal tract is actually the what do you call it, the sphincter that is going to move food from the stomach to the duodenum, and that is that is only moving it about 3 ml per minute per 3 ml per minute. The stomach empties in 4 hours, but an increase in the fatty chyme entering the duodenum can increase the time to 6 hours. So the more fat and the more protein you eat, the slower the emptying of the duodenum will be. Carbonate, why? Because more fat and more protein in the stomach would mean that the chyme is actually more acidic. And the more acidic it is, the slower it will get into the duodenum. The carbohydrate rich chyme will move quickly through the duodenum because the carbohydrate in the stomach does not stimulate severe production of acidic gastric juice. Protein is actually very acidic, and it is going to be slowing down the movement of chyme into the duodenum. This actually is showing you the uh, peristaltic waves in the stomach. First, you have propulsion. Propulsion is the peristaltic wave move from the fundus towards the pylorus. Then you have the grinding. Grinding is the most vigorous peristalsis and mixing action that occurs close to the pylorus. The pyloric end of the stomach acts as a pump that delivers a small amount of chyme into the duodenum. Then you have the retropropulsion. This is the peristaltic wave close to the pyloric valve, forcing most of the contents of the pylorus backwards into the stomach. So there's going to be a backward motion right there. And this is also like the neural factor that would pretty much let you know exactly how the stimulus that's going to result in the emptying of the stomach. Well, for instance, the initial stimulus here is the presence of fatty and hypertonic acidic chyme in the duodenum. And this presence here of the fatty and acidic chyme will stimulate the duodenal enteroendocrine cells and the enterogastrons, which is the secretin and cholecystokinin that is being produced by the liver. And that's going to result in the contractile force of the stomach and would decrease the contractile force of the stomach and decrease the rate of stomach emptying. This is when you feel uh, you don't want to eat anymore. And chemoreceptors also and the stretch receptors would trigger the enterogastric reflex via the short reflex, via the enteric neurons, okay? Also via the long reflex, via the CNS centers, it will increase sympathetic activity and decrease parasympathetic activity, and that would result in the slowdown of the stomach action. So, vomiting. Vomiting is also known as emesis, and it's caused by extreme stretching. Intestinal irritants such as bacterial toxins, excessive alcohol, spicy food, certain drugs can actually cause vomiting. Chemicals and sensory impulses stimulate emetic center of the medulla. Excessive vomiting can lead to dehydration and electrolyte and acid base imbalance. We're now going to start with the liver, the gallbladder, and the pancreas. You have the liver, the gallbladder, and the pancreas. They are accessory organs associated with the small intestine. The liver, the digestion function is the production of bile. The liver is actually very important in our nutrition, but in the digestion of food, the function of the liver is just to produce bile. Bile is a fat emulsifier. When you say fat emulsifier, it remember that fat and water, oil and water, do not mix. So if oil and water do not mix, then that means that if we're going to eat fat, we're not going to be able to digest fat because our enzymes or the enzymes that is available in a gastrointestinal tract that could act on fat to break it down is water soluble. So anything that is water soluble needs to act in the presence of water. How can an enzyme that is water soluble act on something that is oily? It can't. It can happen. So what's going to happen is that your fat needs to be emulsified by a substance that will allow it to be acted upon by a watery substance, and that is your bile. So the gallbladder, on the other hand, is a storage organ only, a storage organ of bile. Then you have also the pancreas, a very important part. It's a very important part in the production of pancreatic juice, or the enzymes of the pancreatic juice, as well as bicarbonate to neutralize the stomach acid. So your liver is the largest organ in the body inside the abdominal cavity. It's not really the largest organ of the body. The largest organ of the body is the skin. Next to the skin inside our body, the largest organ is the liver. It weighs about 3 pounds. It consists of 4 primary lobes. The right, you make sure that you know this in whenever you get into the lab, you have a picture or a diagram picture of the liver. You must know the lobes of the liver, the right, the left lobe, the caudate, and the quadrate. So right here, okay, this is the right lobe. This is actually, you are actually looking on the underside of the liver. You know you're looking at the underside of the liver because you are seeing the gallbladder. You cannot see the gallbladder if you're looking on top of the liver, because the gallbladder is under the liver. So this is the right lobe and this is the left lobe. And because this is the underside of the liver, this top part really is the back of the liver. And you know it's the back part of the liver because you have the inferior vena cava. Because the liver is actually in front of the inferior vena cava. So this is your inferior vena cava. This is the front side, okay? And between the inferior vena cava and the left lobe, you have the caudate lobe. Now, in the front side, right here, between the right lobe and the left lobe, you have a tiny lobe, which is the quadrate lobe. Call it a quadrate lobe because if you're going to see it in reality, in real liver, it looks like a quad. It has what you call it 4 sides. The caudate lobe is caudate because it's at the back of the liver. The left lobe is on the left side of the liver. The right lobe is on the right side of the liver. So in nomenclature, it's really the lobes of the liver are very easy to identify. Okay, the falciform. The falciform ligament is pretty much this part here. It runs also in the top and in the top part and in the underside of the liver. It separates the larger right and smaller left lobes of the liver. It suspends the liver from the diaphragm. You cannot see the falciform liver on the top because you are looking at the bottom of the liver. The round ligament. The round ligament is actually continuous from the falciform ligament. It is the remnant of the fiddle umbilical vein along the free edge of the falciform ligament. And I don't know if you could see this. There you go. That's actually your round ligament there, and that is the falciform ligament. The lesser omentum anchors the liver to the stomach, and you cannot see much of the lesser omentum here because it's been removed. The bile ducts are the following. The bile ducts start as the big, common hepatic. You don't common hepatic duct leaves the liver. The cystic duct is there you go. You've got the cystic duct, which is actually the duct that connects the gallbladder to the common hepatic. That big fat area there is a common hepatic. You see that common hepatic? That's the common hepatic right there. So the cystic duct is actually the duct that connects the gallbladder to the common hepatic. The bile duct, the bile duct is this area here. That big duct there that's been cut is formed by the union of the common hepatic and the cystic ducts. Make sure that you know this, guys. In the microscopic anatomy of the liver, you'll find out that the liver lobules are divided into hexagonal structures. And these hexagonal structures, which you call lobules, are the functional units of the liver. So one lobule is one unit, one functional unit of the liver. And one hexagonal lobule, in the middle of that, you have a vein. You call that the central vein. And then dividing it to help form the hexagon is actually the connective tissue septum. So one hexagonal unit right here is called a lobule. Now this is a high power portion of the liver showing you a the venule. So this area here is part of one lobule. Okay. Now this diagram of the lobule, this is one lobule right here, shows the central vein. There you go. That's a central vein that's located in the longitudinal axis, and also the portal vein, the portal triad. And the portal triad is this. You call it a portal triad because it is made up of the three tubes. You have a vein and an artery and a duct, a bile duct. Bile duct, vein, artery. Bile duct, vein, artery. So each corner of the hexagonal lobule will have a portal triad. Will you find that in systemically in histology? Well, a well-cut liver section would show that. But we are not always lucky looking at a perfect lobule with all of the corners showing a portal triad. But you should be able to locate that in the histology of a lobule of the liver. Okay, let's take a look at the bile composition. Okay, bile composition and enterohepatic circulation. Bile is yellow-green alkaline solution that contains bile salts. Bile salts are cholesterol derivative that functions in fat emulsification and absorption. And bile salts, they travel biles as now when fat is actually being digested, okay, um, bile salts are secreted into the duodenum first. You can see the bile salts that's actually being secreted, that is stored in the gallbladder and is secreted into the duodenum, which is actually the first part of the small intestine. As the bile salts travel through the small intestine, they would allow the lipid digestion itself because the bile salts will hold on to the fat and they emulsify the fat. 95% of the bile salts are reabsorbed again by the ileum, the last part of the small intestine. So bilirubin is a pigment that is formed from heme, and pretty much uh, bacteria break down in the intestine, is actually it's actually converting this bilirubin into stercobilin, and it gives the brown color of our feces. Cholesterols, triglycerides, and phospholipids and electrolytes pretty much uh, are the ones that are actually being acted upon by bile. The enterohepatic circulation is the recycling mechanism, this part here, the recycling mechanism that conserves the bile salts. So bile salts are reabsorbed into the blood, right in this section here, by the ileum, which is the last part of the small intestine. It is returned to the liver, it's returned to the liver via the hepatic portal blood, and it's actually resecreted again, and the newly formed bile, about 95% of the secreted bile are recycled, so only 5% is newly synthesized each time. So there's actually a good, pretty much like a recycling mechanism here. We are not wasting these bile salts as they are actually, so that our liver does not need to be producing too much of it. So these are just showing you the homeostatic imbalances of the liver. One of them is what you call hepatitis. It is usually a viral infection or can be caused by drug toxicity or wild mushroom poisoning. Then the next one is cirrhosis, which is a progressive chronic inflammation from chronic hepatitis or alcoholism. Then you have this is actually where in cirrhosis the liver becomes very fatty and fibrous, and it will result in what you call portal hypertension. Whenever it progresses, liver transplants pretty much would be needed. And most liver transplants, if it will happen, are always successful. But livers are not uh, are not easy to find. They are scarce. But the liver can regenerate to its full size in 6 to 12 months even after 80% has been removed, which means you can pretty much like uh, get a transplant of, let's say, a quarter of a liver only, and it is still going to be good enough for you to survive. The gallbladder is a thin-walled muscular sac. That's your gallbladder there, and it's filled with gallstones. The function of the gallbladder is to store and concentrate bile by absorbing water and ions. Gallbladder contains many honeycomb folds that allow it to expand as it fills. The muscular contractions of the gallbladder would release bile via cystic duct, which flows into the bile duct. Gallstones, which is called also known as biliary calculi or calculus, caused by too much cholesterol or too few bile salts, can obstruct the flow of bile from the gallbladder and becomes painful when gallbladder contracts against sharp crystals. Um, One homeostatic imbalance here is actually obstructive jaundice. Obstructive jaundice, the blockade here can cause bile salts and pigments to pretty much like build up in the blood, resulting in um, jaundicing or yellowing of the skin. It can also be caused by liver failure. How do you treat gallstone? Well, there are now crystal dissolving drugs, or you can actually like break it down by ultrasound vibration and laser vaporization, or even removal of the gallbladder, which is actually probably one of the easiest. Well, actually, the easiest is actually lithotripsy, which is ultrasound vibration. But if that's not possible, then you have to remove the gallbladder. Then we get into the pancreas. The pancreas is one of the most important accessory. The pancreas produces the pancreatic juice, which is actually bicarbonate rich digestive juice that is um, pretty much like secreted into the duodenum in order to neutralize the chyme. And here you could see a diagram of one particular uh, sinus of the pancreas. This is a duct. This is actually the cells that produce the pancreatic juice. And each acinar cells would secrete enzymes, and they would be secreting zymogen granules. And you could see how it looks like, okay? And it releases its secretion right here, and then into the pancreatic duct, and then into the duodenum. This is actually a picture of the pancreas with the acinar cells. You could see that. This is actually the pancreatic juice, and this is actually the pancreatic duct. Make sure that you know how to identify the acinar cells. And there's another portion of the pancreas that you need to know, which is actually the islets of Langerhans. That one is not involved in digestion. The islets of Langerhans is part of the endocrine function of the pancreas. So, pancreatic juice is about 1,200 to 1,500 ml per day and is produced containing watery alkaline solution with a pH of 8. And this is actually produced to neutralize the acidity of the chyme coming from the stomach. And electrolytes, primarily bicarbonate and digestive enzymes, are also part of the pancreatic juice. There's also proteases. It carries also proteases, which are enzymes to digest proteins that is secreted in their inactive form. Proteases are inactive, and pretty much in this will help it to prevent self-digestion of the walls of the intestine. It also will contain amylase, which is the enzyme to break down carbohydrates, lipase, enzyme to break down lipids, and nucleases, which is the enzyme for nucleic acid. Proteases are secreted in an inactive form. They are activated after they reach the duodenum. Now, the enteropeptidase, these are enzyme that is bound to the plasma membrane of duodenal epithelial cells, and they activate the pancreatic protease trypsinogen, activates it into trypsin. Do we have it here? Trypsinogen right there is actually converted into trypsin. So trypsinogen is inactive. It becomes activated into pepsin by the enteropeptidase, okay, that is actually produced by the membranes of the duodenum. In the same token, you have also, once trypsin is activated, trypsin now is going to be activating the conversion of another inactive enzyme, the chymotrypsinogen, into chymotrypsin. And trypsinogen also is going to be converting another inactive enzyme, which is procarboxypeptidase, into carboxypeptidase. And these are now the trypsin, the chymotrypsin, and the carboxypeptidases are the ones that is the active enzymes. The pancreas is retroperitoneal. It is outside of the peritoneum. It is not inside the body cavity. It is deep to it is deep to the greater curvature of the stomach. The head is pretty much like encircled by the duodenum. The tail would be abutting the spleen. So it's very long. It's on the other side of it's on the left side of the abdominal cavity. The exocrine function of the pancreas is to produce pancreatic juice. The acini of the pancreas is a cluster of secretory cells that produce zymogen granules. I mentioned that already. And the ducts would secrete the pancreatic juice to the duodenum via the main pancreatic duct. That's actually your main pancreatic duct. Okay, and the smaller duct cells that produce water and bicarbonate. The endocrine function of the pancreas is to produce insulin and glucagon by the pancreatic islets of Langerhans. Now, um, the bile duct, that's your bile duct, and the pancreatic, the main pancreatic duct, and the bile duct will fuse together in a bulb-like, right there, bulb-like structure, which you call the hepatopancreatic ampulla. And the ampulla opens into the duodenum, pretty much. It opens into the duodenum, and it's actually controlled, its secretion is controlled by the hepatopancreatic sphincter or the sphincter of Oddi, or Oddi. And this is actually the one um, that would control the production or the secretion of the pancreatic juice into the duodenum. So, the regulation of bile and pancreatic juice secretions. Do you think that the pancreas is just going to be releasing substances there for nothing? No, it is actually still regulated. And the regulation actually is actually via the endocrine hormones, CCK and secretin. CCK is cholecystokinin. CCK and secretin are secreted by the duodenal endocrine cells, and CCK release is stimulated by protein and fats in the chyme, which means if you have eaten food that's high in fat and protein, once it reaches the duodenum, then you are going to be stimulating the release of CCK there. Secretin release is also stimulated by the acidity of the chyme. Both CCK and secretin enter the circulation and can cause pretty much the following events. First of all, it causes what you call pancreatic secretion. CCK induces secretion by the isomer cells of the enzyme-rich pancreatic juice. Secretin causes secretion by the duct cells of the bicarbonate. Uh, the vagus nerve could weakly stimulate these reactions during cephalic and gastric phases. The next one is actually bile secretion. Bile secretion by the liver is actually releasing, right here, showing the release of bile salts. Bile salts return from the enterohepatic circulation and are more powerful and it's a more powerful stimulus for bile secretion. Secretin is a minor stimulus only. It can also cause gallbladder contraction. CCK and secretin cause gallbladder contraction. CCK causes the gallbladder contraction itself. The vagus nerve would stimulate weak gallbladder contraction during the cephalic and gastric phase, but secretin causes gallbladder contraction. And finally, it also will result in hepatopancreatic sphincters relaxation. CCK causes the hepatopancreatic um, sphincter to relax. Bile and pancreatic juice would now enter the duodenum. So whenever this is open, it will allow pancreatic juice and bile together to reach the duodenum. There's another opening, there's another duct, which is the accessory duct that is on top of this. It is not controlled by any sphincter. It just throws in bicarbonate rich um, pancreatic juice in order to um, neutralize the chyme. But once, but once you have chyme here that is rich in fat, and you will need bile, then you would need the help of what do you call this, the sphincter of Oddi, to open up, to be opened up, and would be releasing both bile and um, pancreatic juice into the duodenum. I will see you, and we're now going to get into the food after this, into the third part of the digestive system.